puzzle. Heterozygote means we have homozygote for the crest allele. So let's have a look at some of your friends' responses. Not everyone has done, very few has done. Okay, let's look at Debbie's example. Okay. First and foremost, Debbie did a good job. She started out by defining what the alleles are. She said let capital N be allele for no crest, small n be the allele for crest. She used big N because no crest is the dominant allele. She then went on to have the heading, parental generation, and below phenotype, genotype gametes. Okay, you do not need to underline everything. Just underline F1 generation and parental generation will do. Okay, save you time. Okay, because I need you to spend a little bit more time filling this in. Parental generation, then we'll be a bit more specific. Parental phenotype, parental genotype, gametes. Here will be F1 phenotype, F1 genotype, expected phenotypic ratio. Follow the template that has been given to you as a guide. Yes. Phenotypic. Did they put phenotypical? I wrote phenotypical. That sounds very funny. Phenotypic ratio. Yeah. Let's look at other things. Debbie included the phenotype. She included the X, which means mating. Then she put the genotype, and she also put an X, implies mating. She then drew two lines branching out from each individual that represents meiosis to form the gametes. She drew circles around the gametes to show you that they are individual cells. Then she did the cross, which is quite neat. Then she wrote the genotypes, and then she wrote the phenotypes. And the ratio, no crest to crest, 4:0. Okay. Is this what you got?
Yes.
Okay, so how can you double check if you've got the correct genotype and phenotype? Okay, first and foremost, here, this line here is the genotype line. Over here we see phenotype. So be very clear which headers you're using, right? It should be genotype, F1 genotype, then you write the lines here. F1 phenotype, then you write the phenotypes in this row. Okay, so to double check, if you ever ask to draw a genetic diagram, just double check out using the square method. This square method is called a Punnett square. Yeah, what? Okay. So I think Debbie performed everything correctly actually. Yeah? So I think it is the matter of what we inserted into this puzzle. Okay? The puzzle okay, so even if I had done this, I will get the same results as Debbie. Okay. So the question asked for, you said a heterozygote. That means one individual will have been this. Then the next one they say is homozygote for crest. No crest. So therefore, you will have been this individual. If you were to just do a quick double check, this is what you would have gotten. I think you would have gotten a 1:1 ratio. Okay? So, but the formatting, largely this is correct. Debbie did not draw all the tables. Okay, you do not need to draw any tables. Please save your time. Okay, let's look at other examples. Okay, this one is black color. Let's see, with white lines. Okay. Okay, also can see. Okay, let big N be the LU for no crest, small n be for crest. She then underline parental generation, parental phenotype, parental genotype. Everything is good so far. No crest made with crest. She wrote the correct genotype. She then branched out meiosis to form the gametes. She then performed the cross. Okay, this is called the crossing part, the genetic cross. This is fertilization essentially. Then the F1 genotype, then the F1 phenotype, and then she wrote the expected phenotypic ratio. Okay, because actually we can create ratio for a lot of things. It could be a genotype ratio, it could be a phenotype ratio, but we always ratio the phenotype because that's the thing we can see. So expected phenotypic ratio, no crest is to crest, 1 is to 1. Yeah. Okay. I hope you find some pleasure in solving puzzles in this chapter. As a student, I didn't, but as an adult, I don't know why I find it quite enjoyable now. Maybe it's old people kind of enjoyment. Hello there, it was. It's back. Oh my god, it's inside! Don't worry, it's just trying to find a home. That's a wasp, by the way. Wasp, by the way, can sting multiple times. Unlike bees. And then there are different kind of wasps. Some wasps make their homes out of soil. Some make their homes out of cellulose. So it's like paper. Yeah. So actually what it's trying to do is just trying to find a hole. You know your table, look at your table in front of the screw part, the metal, you see those metal screws, you come with a hole. Yeah, wasps like to make their home in there. Don't know why. Oh, the smaller ones. Don't know why they enjoy to put their home in there. Yeah.
Is it normal for these bees to come into a hole? I get them like almost every day.
Oh, maybe you have something inside you want. Okay, this is Rayhan's work. Rayhan, just a broad scan. I feel like she has most, all of the pages on the left hand side. Just remember to underline the parental generation and F1 generation. Rayhan, you also have to define at the very top what your LUs are. But then that, if it had been defined, I think it's also well done. Okay? So the probability for the teacher getting no crest will be 50%. If you are given an MCQ, do not draw this diagram. Okay? Just draw the square. It saves you a lot of time. If you are very scared that you get the wrong answer after drawing this diagram, especially last time as a kid, when I do this crossing part, right, chaos ensues. Yeah? So if you're very scared that you cross the wrong thing, just go and draw this square. Then you can double check. There's always a way to double check. And therefore, the only dangerous part is what genotype you put in as the parents. Right? Because everything else you can do it. It's only the parental genotype that you need to put in correctly. For those that have not tried, I really encourage you to try. This one with more practice you get better, like graph drawing. Okay? Today we're going to try other puzzle examples. 2 more scenarios. 1, 2, 3. Okay, may I get you to go to, yeah, pause your assignment. Oh, I meant Rayhan, okay. Well, it's not your Rayhan. Okay. Okay, may I get, wait. Oh, this course. Oh, thank you. Okay, now may I get you to go to part B? we're going to try some other puzzles that you can really see in real life. I've already introduced blood groups to you. So we're going to try solving puzzles using blood groups now. What's the thing? Oh, how to draw a Punnett square, is it? Just draw a square, then draw across. Yeah? Now let's try puzzles using blood types. First and foremost, we do a quick recap. Can you identify which are the alleles we use for blood typing? By the way, this is convention. People always typically write the alleles in this way. Yeah, so you don't need to go and come up with your own alleles. Remember what these alleles look like, because this is what typically normally people use. Which one out of these four is not an allele? Okay, AB is actually doesn't exist. Okay, so let's try a puzzle using blood types. Okay, the puzzle goes like that. Draw a genetic diagram to show the possible blood groups of the children of a father with heterozygous for blood group A and a mother with blood group AB. I still need you to define the alleles, even though it is quite convention the alleles people use. So, and define it like that. Let IA, IB, IO be the alleles for blood type A, B, O, respectively. Then you can continue your puzzle. Okay? Okay, can everyone try this puzzle? Then we will just double check with each other what's the probability you get. Sorry, what are all the possible blood groups? Take this as a practice. What would the genotype of the person be if this person is heterozygous for blood group A? Oh, okay. Yes. So phenotype are characteristics. You may not be able to see it, but there are still things that appear on the surface. If you're done with your genetic diagram, please let me know. I'm going to come around to have a quick look. I'm going to make sure you all can draw this well. if you want to double check your answer, the individual is heterozygote for blood type A, mate with an individual that is blood type AB. So you check your ratio. There should have been a blood type A is to blood type okay, if you did not write you write blood type. Yeah. Be a bit diligent. Because, okay, think about it, it's very funny if you tell someone that the phenotype is A. Okay, your phenotype is alphabet. Blood type. Blood type lah. Okay, your phenotype is a blood type. You cannot be A, B, or O. Okay, blood type. I think it was an AB. Okay, so the ratio we have got something like 2 is to 1 is to 1. This should have been your N. Okay. If a particular blood type or particular phenotype does not exist, you don't need to include it in your ratio. So no blood type O, don't need to include now. Yeah, I hope you're getting more comfortable drawing genetic diagrams. We'll try one more example. I'm just going to do a quick scan as I move across. I've already looked at quite a number of your work. Goodness. We've learned several things. We learned that alleles can be dominant over each other. Okay. We learned that alleles can be recessive. Okay. We've also learned that alleles can be co-dominant, as in the case of blood type. Because you have an IBIA, you end up getting blood type AB. This is outside your syllabus, but last week, I think it's with your class, we talked about incomplete dominance. And incomplete dominance would be like a pink cow. versus if red and white exists, it will be red and white patches. Okay, that was last week. I'm going to erase this because it's out of your syllabus, so actually you will never need to write incomplete confidence. So far, the examples we've been playing with are with alleles that are present on what we call autosomal chromosomes. How many chromosomes do we have in our body? 23? 46. We have 46 chromosomes, 23 pairs of them. Right? So what do I mean? This is pair number 1. Oh goodness. 3, 1, 2, 3, 4, 5, 6, 7, 8. These are homologous. 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Okay? We have 22 pairs of what we call autosomal chromosomes. I specifically say autosomal because actually the last pair, they're not exactly homologous. They define your gender. It is the X and Y. Y is the shorter of the two. You could have 2X, that would make you female. Or you could have 1X, 1Y, that would make you male. The Y chromosome is way shorter than the X chromosome. They are not homologous. Okay? Homologous means that they will have the same genes. Could be different forms of the allele, but the same gene. For example, this person's blood type could be, if I look at one of the autosomal chromosomes, at these 2 points over here, it could have had the IA allele and the IB allele. This person's blood type AB. Okay? But these 2 chromosomes over here, the one that define your gender, they're a bit special, these sex chromosomes. These sex chromosomes do not have the same genes in each one. I mean, that's why we can come out different, male and female. And you look at the Y chromosome, it's much shorter, right? That means to say, whatever the X chromosome has in this region, like over here, all these genes over here, the Y chromosome confirm won't have all these genes in that region, because it's way shorter. In other words, the Y chromosome does not have some of the genes that the X chromosome had. It is just a shorter strand of DNA. Okay? But we have a problem. Hey, what? I just realized I'm talking to a background music. Quite nice, huh? I should switch to epic music sometimes. Okay, so, but here's the interesting thing. These chromosomes, sex chromosomes, actually have genes on them too. In fact, one of the genes that can be found on sex chromosome is the one that defines whether you can see color. Okay? And it can be found on the X chromosome, not the Y chromosome. So whether you can see color or not, that gene is not found on the Y chromosome, it's found on the X chromosome. When certain genes are found on sex chromosome, we say that it is, so this gene here is sex linked. Because it's linked to a sex. But we have a problem though. Because these sex linked genes, right, they also do follow the rules of dominant recessive. Where, okay, I'll show you the problem now. Let's say, okay, this is real. Color blindness is a recessive value. Okay, I'm going to say let small n. Let's start with big N first. Let big N be the allele for normal sight. Then let small n be normal color. Let small n be the allele for color blindness. This this is real, color blindness is a recessive allele. But guess what? It is found on the X chromosome. And found where? It's found somewhere up here. Okay, then I present you the issue. Let's say someone is female, that means 2 copies of X chromosome. The alleles could be big N, right? This lady would be, can see color, right? Okay, so can see color. Let's say we have another lady, 2 X chromosomes, 1 big N, 1 small n, heterozygous for the gene, or for the allele. This person will be, so you can see color. Okay, so you see everything is still quite logical, whatever we've learned. In fact, if the lady has 2 copies of the recessive, then it will be colorblind. But here lies the issue with males. You see the issue with males? Okay, let's look at males. In males, X and Y, right? X chromosome and Y chromosome. Let's say the male has the dominant allele. You will be able to see color, right? But the moment the male has 1 copy of an X chromosome, then has the recessive allele, will be colorblind. The laws of recessiveness are almost a bit no point here, because you just need 1 copy and the male can be colorblind already. There's nothing in the Y chromosome, which is shorter, to mask over this recessive allele. So in reality, actually we find a lot of individuals that are colorblind tend to be found in males. We laugh. Okay, so a lot of males, if you want color blindness, then we found females a little bit more in proportion. Because females need to have 2 copies, then they will be colorblind. Male just need 1 copy. Therefore, if you think the probability, if there is a mother that is colorblind and a father that can see color, what's the probability that the boy that is born from them will be colorblind? How many percent? What? 93 point? 33 point? Okay, let's look at this puzzle. If a mom is colorblind, what does it mean? She has both X chromosome will have the N, right? Okay, sorry, small n, right? Okay. The father would be just 1 big N. To be a boy, you need to take 1 Y chromosome, right? Confirm must take from the father. The mom must take 1 X chromosome from mom. Cannot take X chromosome from dad, correct? You take from dad, then where you take the Y from? Okay, so you take from dad and from mom. So what does this mean? Now, any boy that is born, 100% will be colorblind. I mean, sad or I don't think it's sad. You can still live life, no? Okay? So, but you see these kind of puzzles we can solve, right? Once you understand sex link. Okay, then how about this? What about a child is a girl? Can you calculate the probability that a girl will be colorblind from such a parent? Okay, so we try out. This set of parents, what's the probability that a daughter born from these 2 parents will be colorblind? Oh, really? 0. Is that true? Okay. So I'm going to replace that. I'm not going to write this symbol anymore, okay? Instead, let's just work with X chromosomes. So this is what you can write up. The mom would be XX. Okay, then you can write superscript. Okay, then you can write, what was mom? Mom is like that, right? Okay, dad is this. Okay, so you were saying females 0% will be colorblind, right? Okay, 8 now. So first, 1/4 of the daughters, okay, good. Still okay. Oh, because all daughters, right? Okay. So anything else will be sons. Okay, yeah. So daughters will not be colorblind. Yeah. So actually, knowing the genetics of this, if you see these two individuals, right? Is it how will you go about family planning? Is it ethical to advise someone to not have kids if you feel like there's a chance that you'll get a trait that you feel that you don't want your child to have? Is that ethical? It's a personal choice. Because right now we're just talking about color blindness. There are other sex-linked genes that can result in things more serious. One gene that's also sex-linked, it's called hemophilia. This particular allele, if a person were to have this disease or this illness, blood cannot clot. Okay, so if they were to get a cut, they will just bleed. But because you learned about hemostasis in JH3, blood clotting, all that process, right? Hemophilia, blood cannot clot. So if you can already identify father and mother, you get these two father and mother come together, right? The probability is as such, right? I don't know, is this something to think about? Right? How my wife advises parents? Okay? With that, let's try. You know an example to try. I want to play with an example. Let's see if we have any puzzles for you to solve. Okay. Your TE, let's see if we have some puzzles to play with the TE. Wow, that was close. I thought I'd open the exam paper. Okay, this is not sex linked. This is sex linked. Okay, let's try this example. Okay, let's kill two birds with one stone. Can you go to question 9 of your TE 8.1? Let's try one example. Okay, practice now. Sex-linked genes. Practice drawing genetic diagram to solve this puzzle. Male with normal color have a father who is colorblind. They are planning to have children. In this puzzle, they provided you a family tree also. Give you a little bit more clues to solve the puzzle. Okay, you are supposed to predict the phenotype and genotype of the children of these two individuals, yeah? So the question mark is the thing you are trying to find out. This got two parts to this question. You must first find out what these two individuals' genotypes are. Okay, two layers to this puzzle. I suggest before you start drawing the genetic diagram, check with your friend whether you managed to find out the genotype of the parents first. A good practice as a student, beginner geneticist, is to draw out the genotypes every time you were to get a family tree. Write out all the genotypes, try to solve it. ask a good question how to define. Okay. Because here we're working with a chromosome that has an allele on it. You could define it as, I give an example, let X and be the X chromosome carrying the allele for colored vision. Then X small n. Is that a shorter way to write this? Let's insert a respectively. Okay. Then Xn that would be a shorter way to write. Don't need to define y. Okay, there's nothing on y. Okay, so you just need to define the allele on the X chromosome. Have you first solved the family tree yet? You know who has what? Is the son's mom. Oh. Did they provide any other details? No. So actually they are wide variety, right? Plus the mom could either be so now could be either or. And how? But can we still solve this? We know that the sun is not colorblind. Therefore, the sun has big N. So Y has to come from who? Y has to come from the father, right? So Y has to come from the father. That means this N over here needs to come from the mom. Okay, but we still have to solve it. But it doesn't matter. Because they are asking you to solve the offspring from these two, not of the parents. Okay, so now this daughter. Okay, do you have a problem? Okay, this one, the mom will have to be. We don't know, right? Okay, so but the mama confirm will have one small n, right? Because came from the dad. So this one confirm have to be. Why must it be big N? Because the mom is not colorblind, right? So it is irrelevant to know whether the mom's genotype. Because the puzzle we need to solve is between these two. To find out no, you didn't have to do anything. Because question 9a just wants you to find out what would the offspring be between this and this. Yeah, so as a student I gave up quite easily. if I encounter something like that, then I'm like, oh yeah, I don't think there. But actually quite fun, huh? You try to solve these puzzles. Sorry, what? Yeah, because got male and female. Mm. If you think you're done with your genetic diagram, just raise your hand, let me know. I can do a quick check for you. I'm checking more of the way you present your answer. I need to check also what the answer will be. affected. Female is to unaffected. Male is to affected male. Oh, sorry, or colorblind. 2:1:1. Okay, so something like that. Okay, as I was walking around, I think largely a lot of you got it. Where X and Y chromosome are involved, may I invite you to write the gender also? Okay, because gender is also a phenotype, whether you're male or female. Any other examples with the color blindness? Don't have. Shall we try another genetic diagram, another puzzle? Number 10. Okay, try another blood type one. Using the symbols, show how two parents may have children of all four blood types. Wow, they don't even tell you the parents genotype now. Now you need to mentally work backwards to solve this puzzle. Yes? Oh, for which question? This one? Oh, so are you asking can I use circle and square? Oh, cannot. Okay? So circle and square are not typical symbols that geneticists use. They use alphabets. Okay? So now let's go to blood type. Okay, in the event that the question has defined the alleles for you, then good news. You do not need to have the definition line. But you're scared, you still write down. Yes. Oh, what is question 9 shall be? Oh, okay. Okay, actually this is a good question. Explain why the observed phenotypic ratio may not be as expected in this prediction. In other words, I ask you, what's the probability that you will have a colorblind male? Probability. Can you calculate? 25%, right? 25%. Okay, this is a matter of probability already. Okay? So I ask you now, if these two parents were to have 4 children, I ask you, if these two parents were to have 4 children, will one child be, will 2 of the girls be definitely unaffected, one of the guys be unaffected, and one of the guys be colorblind? Why not? Yeah, okay, I don't know the math of this. But this is all due to probability. Every time one kid pops out, right, there is a 25% chance it will be male and colorblind. That probability could be, this couple could even have the chance of every child they get off, you just get the 25%. Make sense? Okay? There's a chance every child that comes out 25% chance, even though it's a 25% chance you will get a male that's affected, it could happen 4 times. Right? Yeah? So first child come out, could be this. Second child come out, also can be this. Third child come out, also can be this. Fourth child come out, also can be this. Then this feels a bit weird, right? Clearly it's going to be 25%, but how come we do not see any ratio? Okay. These ratios only make sense if you have like thousands of kids. Okay? Okay? So let's say you have thousands of kids, and then you know what happened? Maybe after the 4th kid, right? Then you start to see unaffected female. Right? Then the 5th kid you see unaffected female. So you only see these probabilities take shape if you have thousands and thousands of kids. Then the ratio will start to really surface to the top. You start to see the ratios appear. But when you only have 1 kid or 2 kids or 3 kids or 4 kids, right? You will not see these ratios And this is what this question is trying to ask you. Why is it the ratio we observe, right? This parent have these four kids, we don't see this ratio, but yet this is what we expected. That's because, okay, I'm just going to show you the answer, because it's always the same answer. Here we go. We got the gist of it, but this is how we usually phrase ourselves. If you go to A-level side, you take biology, this question will often come up again. They like to ask you why what you observe is often not what you calculate. It's because you have not reached a sizable number yet. If there's one vocabulary that I find very useful to explain this, it's about sample sizes. Your sample size is too small, so you will not see this ratio appear. I don't know if this was taught in math.
So we actually find out how to calculate the probability.
Oh, okay, you all know how to calculate probability. If this didn't quite make sense, I think a better example would be on gender. You know that it's a 50-50% chance whether you can get a boy or girl, right? Yet, you can see some families, everybody in the household girl, or everybody in the household boys. Even though it's a 50% chance, you do not see that ratio. You can only see the ratio if this couple have thousands of kids. Then you see around 500-ish boys. The other 500-ish are girls. Okay? Are you planning already? Oh, okay. No, then the question should be when's the earliest, right? Oh, where's the latest? Okay. The latest is usually around 45. Yeah. 45 is pushing it already. Roche is aiming for 36 kids.
Not in this economy.
Sorry, your 36 kids starting what age? 18. How old are you this year?
Yeah, I don't get pregnant that young.
Oh, okay. 36 kids.
I heard like that, yeah.
find somebody. I thought you already. Oh.
We need to find somebody who can handle this.
Okay. When you're at the end of this chapter, I'm going to close up the chapter today. Okay. Why study genetics? We study genetics because at the end of the day, it is what defines the differences that you see among us. In biology, we say that there is variation. You've seen the word variation before, right? Genetic variation, in fact. We study genetics, how we inherit all these alleles, because we want to understand the variation we see. Why is it some of the variation we see is distinctly either this or that? For example, you're either this blood type, or this blood type. You either have the allele, wait, you don't check up. Can you see if you have this extra pendant coming out? It's also defined by just a single gene. Can you see? Who don't have one? I'm not sure you don't have, is it? Okay, if you do this, you catch, can you see this pendant appear? Who doesn't have it? Okay? These are also defined by a single gene. Okay? Other things that can be defined by a single gene, whether you can see in color or not, right? This kind of variation, right, is very clear cut. It's only yes or no. Okay, we're going to watch a video on the kind of variation that exists in this world.
You will immediately observe how cute and fluffy they are. But you will also
Hey, what the?
that they are different.
Oh my god.
They vary. Yes, some are puppies and some are kittens.
Okay, let's start again.
Look at these cute baby animals. You will immediately observe how cute and fluffy they are. But you will also notice that they are different. They vary. Yes, some are puppies and some are kittens. There is a great deal of variation between species, but also there is variation within a species. One puppy is fluffier than the other. They have different coloured fur, and the kittens have different coloured eyes. Variation is the differences in the characteristics of individuals in a population. It can be due to genetics, the environment, or a combination of both. Let's start with genetics. Different members of a population have different DNA, unless they are identical twins. This means they have different versions of genes called alleles, and these alleles give rise to different characteristics. For example, on the eye colour gene, this kitten has an allele for blue eye colour, whereas this kitten has an allele for brown eye colour. If the species reproduce by sexual reproduction, the random mixing of alleles from both parents results in extensive genetic variation within the population of a species. This means that some individuals are better adapted to their environment than others, allowing natural selection to take place. For example, in a population of rats, some will either have better eyesight than others. These will be able to avoid being eaten by predators, so are more likely to survive to reproduce and pass on this advantage to their offspring. If you want to learn more about natural selection, watch this video. Some examples of variation in humans that are controlled by genes include eye colour and blood group. If you have a blood group A, it's because you have the alleles for this characteristic. This is genetic variation, and it is something that you are born with, and nothing can change it. Some variation is due to the environment or lifestyle of the organisms. Examples in humans are hair length. It depends on how short you decide to cut it. Your accent, as this is linked to where you live, and if you have any piercings, tattoos, or scars. These are characteristics that you acquire throughout your life. Many characteristics are due to a combination of both genetics and the environment. For example, the height a person can grow to is determined by genetics. Tall parents tend to produce tall children. However, eat a healthy, balanced diet. Also, there is some debate on how much intelligence is due to genetics or the quality of education the person receives. Is intelligence more due to nature or nurture? To answer these questions, scientists can do experiments using identical twins. If the characteristic in both twins is the same, then it is most likely due to genetics. If it is different, then the environment has played an important role. For example, identical twins will have the same eye colour, as this is only due to genetics, but they will have different personalities, talents, likes and dislikes, and this shows that these are also influenced by the environment.
Twin studies are very often done to really decide or figure out whether a certain particular characteristic is defined by genes. In our lives, you notice that some characteristics have a lot of variation. Yet some have only a few kinds of variation. I'll show you an example. When we look at height, if I were to plot a graph of the, let's say this is the number of individuals, and here is the height, where if you go further out, this is 0, height 0. And then further out means taller. You find that in the population, we have very few individuals in this region, we have lesser, but we find majority of the population tends to be in this range of height. We have some extremes, where some individuals are extremely tall, some extremely short, but a lot of the population are in this region. Okay, let's say for one gender, maybe male. We say this kind of variation is a continuous variation because we do not see any clear-cut variation. There is a range of different heights. This is a bell curve. Yet some form of variation are very clear-cut. For example, eye color. You are either, or blood group, you are either A, B, O, or AB. Yes, within a population there may be different numbers. Okay, but O is the most. But what you find is that it's very clear cut. It's either A, B, O, or AB. In this example, we have people who are of the height of 140, and then some are 140.1, 140.2, and then some over here 160, and so on and so forth. It is continuously varying. But some characteristics are very clear-cut phenotypes. Okay? So the reason why I'm introducing this to you is because there are two kinds of variation. Some are discontinuous, some are continuous. Can you think of other continuous variation that exists among our population? Any other thing that varies continuously? Like you cannot quite see different clear types. Perhaps the color of our skin. There is no clear different color, but there is a range. Yeah? Anything else? The volume of your voice. Or could that be trained? So height, I like height as a very good example. Because really we have a continuous variation in our population. Okay, but here's the thing. Very often, variation that are discontinuous in nature actually are defined by just usually a single gene. Usually one or two genes only, maybe single or two. But things like height that has a continuous variation, actually they are defined by many different genes. Because if you think about it, something like height requires a lot of things. For example, you may require bone strength. Right? If your bone not strong enough, you cannot possibly grow very tall. It could be about the different kinds of hormones you produce. How much hormone you produce. Right? A lot of things contribute to height. What else could possibly contribute to height? It could be about the muscle strength, right? How much muscle you can produce in your body. Many different genes contribute to height. And therefore, it is the combination of what alleles you get that will define how tall you get. Imagine if at every single gene you just get the version of it that will result in shortness, then you'll be this individual over here. Okay? But if you get the form of the allele that results in tallness for every single gene, then you'll be this extreme. But if you get, and so you can see a combination of different alleles and genes will result in you falling on different parts of this continuous variation. But for characteristics that are just defined by a single gene or one or two, you can only fall within certain categories. That's the difference between discontinuous and continuous variation. And in reality, very few traits are discontinuous in nature. A lot of things we see in ourselves are continuous in nature. To end off, another interesting thing about continuous variation is that it can be affected by the environment. Give an example. Even if you have the alleles to have strong bones to grow tall, if you don't take in enough milk, enough calcium, you're never going to reach your full potential. That's how the environment can also affect you as an individual, or whether you can achieve your fullest potential, genetic potential at least. Therefore, all the genetic puzzles you are trying to solve are all discontinuous variation. Okay? It is impossible to solve a puzzle regarding height, because there's just so many kinds of height. What's the what? Oh, I don't know what's the widow's peak. I forgot what it is. What is it? Oh, it's the hair. Okay, Ma Rongxuan asks. Ma Rong. Okay, she asks what's widow's peak. You look in your textbook. You all go and check your pen now. A widow's peak is a hairline thing. Hey, go and see. Someone with a widow's peak will have this pointiness at the head. It's dominant. Who? Mr. Nick has. Okay, everyone pull back your hair now. You guys see you have a widow's peak. That's dominant, huh? Hey, you have, Crystal? Oh, that also has. Y'all go have a look. Okay, anyone don't have, then since it's dominant, right? There's a chance that we may see it more. If you don't have, that means you have 2 copies of the recessive value. Do I have? I don't have. Wow, 2 recessive. Okay, but what does this mean? It is a misconception to think recessive values are bad. Doesn't mean I'll get bored. Hey, don't try and curse me. Okay. Recessive values are not bad per se. Okay? It really just is an idea of whether it will be masked or not. Because you look at this, whether I have this or not doesn't affect my life, right? It will not affect my level of survival in this wilderness. Okay? Okay, so I want to remind you, dominant recessive has nothing to do whether it is good or bad. You can have diseases that are dominant. Some diseases are recessive. So it's really just about whether they can mask each other or not. Is cancer down to genetics? A lot of studies have shown that yes. So you could have genes that predispose you to cancer. Okay, alright, that's all. We can cut here today. Go for a recess. Okay, thank you, class.